and it's a delight to have Jim here to talk about that.
Thank you very much. Well, thank you so much for that really kind introduction. I was very flattered to be asked to give this lecture. In fact, I wore one of my favorite DNA ties, and if you play your cards right, I won't show you my DNA tattoo. But seriously, Tom is a giant in our field, and I've known of Tom ever since, of course, I was in medical genetics. And it occurred to me today, as I had these wonderful visits with all kinds of people, both yesterday afternoon and today, that the department here of genetics really reflects and embodies two things that Tom reflects and embodies at the individual level. And that is, on one hand, rarefied science and just superb clinical activities in conjunction with really kind personalities and good humans. It just, that really shines through, and it's been such a pleasure to get to spend a couple days here, and such an honor to get to give this lecture. I wanted to start by just talking a little bit about Tom. He is, a true triple threat, what everybody has looked for in academics. Someone who can teach, someone who can do research, and someone who can see patients at the highest level in all three of those things. His pedigree is unbelievable. I mean, from Oberlin College to Oxford as a Rhodes Scholar, to Harvard Medical School, to the National Institutes of Health, to the University of Washington, I have that down twice, to Yale, to then the University of Michigan since 1974, right? And I should just mention in that context that I think oftentimes when people are at a particular institution, they actually sometimes don't recognize the illustrious reputation that they have worldwide. And the University of Michigan is thought of so highly and so respected all over the world in this field. He's been an educator and a mentor beyond just to the many individual people whom he has mentored and taught, but really to all of us. He and David Ginsburg and Francis Collins with their classic textbook on medical genetics. He's been obviously a researcher who has made seminal contributions to our understanding of hormones and mechanisms of action. And finally, as a physician, he clearly is somebody who has shared his wisdom and his compassion with people for decades and has made a huge difference in people's lives. So it's a huge honor to be able to give this lecture. So turning from Tom's genome to all of our genomes, this is a sample. It's 1/1,000,000th of the human genome, and I show this to us all to remind us of just how big the human genome is. It is huge, and that is what confronts us and what presents most of the challenges inherent in harnessing it, in trying to figure out and trying to apply its analysis to individual patients. And it's so big that I could show, every second, I could show a slide like this, and we would still be here in 12 and a half days, and we would have only gotten through the haploid portion of the human genome. It's sheer size and the fact that the important bits are oftentimes surrounded by things that we either don't understand or probably really do represent excess baggage that make it so challenging to figure out. Now, we all know that the genome is interspersed with genes, or more accurately, fragments of genes, that is, exons. And that we have polymorphisms, both polymorphisms that represent single nucleotides as well as copy number variants, giving us some degree of variability from the people sitting next to you, although, of course, we should also remember that we are 99.9% the same as well. Many of those polymorphisms are meaningless. As far as we can tell, they neither affect things at an evolutionary scale or at a clinical scale. Some influence traits, like the color of our eyes, or medically important characteristics. And then occasionally we find rare mutations, although I shouldn't really use the term mutation anymore, I should say pathogenic variants, that greatly influence one's health. In the midst of this, right, in the midst of this huge genome that we're now beginning to be able to glimpse on a relatively routine basis, I think it's worth pointing out and emphasizing that family history has never been more important, that it has both diagnostic and screening utility in the clinic. It's an important diagnostic tool for one main reason, and that is that what do we do when we see a patient, right? The first thing we do is try to make a diagnosis, and in the service of trying to make a diagnosis, we generate a differential, right? We generate a list of things that may account for our patient's problems. And if we can determine through family history that a patient is likely to have something that is genetic, that can greatly winnow our differential diagnostic list. So it's a huge aid in diagnosis, and it's also an effective screening tool in that about 1% to 2% of those in the general population carry a pathogenic variant that strongly predisposes them to a serious but preventable disorder, just the kind of thing you want to screen for. So it can be an important clue in illuminating the risk to such an individual and therefore executing preventive action in them and in their family. So it's really never been more important. It also critically informs the interpretation of genomic data for patients. So what we're learning as we suffer under this deluge of variants that we're now being subjected to because of extensive sequencing is that going back to the family history is oftentimes the most important and most accessible clue that can help us figure things out. It also helps us understand the genome with regard to basic human biology. So we haven't always had this tremendous ability to analyze the genome. We have had the ability to sequence DNA for a long time. For decades, we've been able to sequence DNA. The problem is that we have had to do that one DNA fragment at a time. And when I was in graduate school, you would spend basically 36 to 48 hours, an experiment that took that long, to, on a good day, get 250, 300 base pairs at the end of that experiment. But massively parallel sequencing takes advantage of miniaturization to carry out simultaneously millions of reactions in tiny little, test tubes, if you will. And then through informatic processing and the kind of technology that allows the phone in your camera, or excuse me, the camera in your phone to work, we can then assemble genomes. We don't even need to assemble genomes anymore because we can now map them to reference sequences very quickly. So you're all sick of this graph, as am I. It shows how rapidly the cost of sequencing has declined. And really the only parallel that I can think of in my life has been consumer electronics and specifically personal computers. I'm both old enough and nerdish enough to remember the Commodore PET 2000, which was the first personal computer. It cost about $3,000 at that time in today's dollars, and yet it has about 8 million times less dedicated memory than my smartphone. And you can also make phone calls with your smartphone and play Angry Birds and all kinds of things. The question, though, is for all of that, marvelous reduction in technology and reduction in price and enhancement in technology, the question is, what good is it? All right? Now, the smartphone is pretty easy to answer. How many people in this room have a smartphone? Well, I should ask, how many people don't, if you want to, if you don't mind? Okay, we've got a couple of proud, people with the old technology. How many of you have had your genome sequenced? Okay, we've got one. Somebody almost raised their hand, so I'm guessing they had their exome sequenced. But, there's a reason for that, right? This is a graph of penetration of smartphones into the US population. And, I would say the penetration of whole genome sequencing into the population has been really low. And there's a reason for that. It's that the smartphone is really useful to us. We're still kind of casting about trying to figure out how the genome can really be of use to us as individuals. And that's really what I want to explore with you today. What are the near to midterm practical applications of massively parallel sequencing? And as a physician, I'm interested in that with regard to human health. Okay, I think we've obviously gained incredible insights into biology, into evolutionary science by sequencing the heck out of all kinds of things, and that's a wonderful endeavor. Oftentimes with the new technology, we're reminded, I think, of the old adage that when you have a hammer, everything looks like a nail. And we've been using that hammer to do a lot of, pounding of nails. My question is for us, what are the appropriate nails for the hammer of massively parallel sequencing in medicine? So the question is in sick people and in healthy people, right? That's what I'm interested in. Well, one of the things that was really fairly easy to figure out from the start would be that massively parallel sequencing will greatly facilitate the diagnosis of entities when they primarily are caused by a genetic lesion, a straightforward, say, Mendelian process. And we know that making a diagnosis is the linchpin of medicine, and it has always been so. It guides prognosis and treatment. It enables general medical progress, and it provides tangible benefit in that sometimes it guides effective treatment, sometimes it affords prevention. It can almost always inform reproductive decisions if you are in a society where you are allowed to exercise that, and it ends the diagnostic odyssey, right? That saves both anxiety and resources. And what I mean by that is, so many of the patients that we see, of course, have gone from doctor to doctor over many, many years and have not gotten a diagnosis. And that's something that is not only is wasteful for the system, frustrating, and sometimes dangerous for the patients because of the, tendency to do invasive testing, et cetera, but it has a psychological effect on people. And I would say that one of the advantages of being able to make a diagnosis is giving people putting a name on something helps people at some level. Now, we would like to do far more than that. We would like to fix them, right? But short of that, even making a diagnosis does have some real benefit and if you will, utility for people. So in sick people, right, this diagnostic tool can now be used on a pretty routine manner. And this is an example of a 47-year-old that we saw with sudden cardiac arrest. She was resuscitated successfully, and on EKG, it looked like she had long QT syndrome. Now, I would get referrals for long QT syndrome for years and really not be able to do much as far as figuring out the molecular etiology because we didn't have the ability to sequence all the genes that were associated with the disorder. The problem is there are many genes associated with long QT, and the clinical conundrum is that knowledge of which gene is mutated can affect both how we treat people and their prognosis. So application of MPS was an immediate, obvious kind of application, and it is now routine to run panels, for example, of genes on such patients to detect the mutation and guide their treatment. But not only can we guide their treatment, of course, we can do what many people who come in are even more desperate for, and that is, how can you help me figure out which of my kids are at risk and hopefully prevent it? And if we do know about something like long QT, we stand a very good chance of being able to prevent sudden death in them through either pharmacologic means or the implanting a defibrillator. Once in a while, you hit a real home run, right, when you make a diagnosis. Sometimes it, guides you to some extent, and once in a while, it dramatically changes somebody's life. So I love showing this slide, not because I want to give the wrong impression that the story I'm going to tell you is the norm. It is not. But because I want to show and demonstrate the potential for making the right diagnosis, and that sometimes it has huge dividends. This is a 36-year-old who at age 6 was diagnosed with hereditary spastic paraplegia. Over the next few years, she really got to where she was confined to crutches if she was going a short distance, or a wheelchair if she was going farther. In addition, she had very painful episodes every day of leg spasticity, and it really severely affected her life. We enrolled her in our study of whole exome sequencing to try to make diagnoses in people who had defied diagnosis through traditional techniques. And of course, over the years, she had been to many, many different places. And on whole exome sequencing, we found a mutation in the GTP cyclohydrolase 1 gene, which was very exciting to us because it had been associated with a condition called dopa-responsive dystonia. And that, even the name of that immediately suggests to you that maybe there's something therapeutic we should do. So we called her neurologist and we suggested, and the neurologist, Jane Fan, was part of our study. She had nominated the person for being in this study. And we said, this is an exciting finding. It looks like maybe DOPA would be a good thing to use in her. The good thing about DOPA, of course, is that millions of people take it. It's a first-line agent for Parkinson's. It's well-tolerated, an oral agent. And she came back 6 weeks after starting DOPA and had left her crutches at home. She was really leading a normal life. It's now been about 3 years. She has had no return of her spastic symptoms or her mobility problems. And that was obviously an incredibly gratifying response. And it was fun to get to publish it, what was also fun was where it got picked up, which was Cosmo. And if you think that like the New England Journal gets a lot of press, Cosmo beats the impact factor of the New England Journal way to hell. So it's there somewhere between Bad Girl Sex and Am I Normal down there. So that was a really, but I want to emphasize that's not the norm. What we hope is it will increasingly become the norm, right? So in our entire study, in which we performed whole exome sequencing on a cadre of about 650 people, all of whom had something that seemed like it might be primarily genetic, but had defied diagnosis with traditional techniques. We found about 17% of those individuals with a slam dunk positive diagnosis, another 20% with a very interesting, perhaps very tantalizing case that this variant was responsible for their phenotype, but not quite enough evidence where we could say it for sure. And in about 2/3 of cases, it was negative. When you break it down into the different categories of patients, you see a fairly broad range of diagnostic success. I'm particularly interested in things like dysmorphology that we get a 37% either positive or possible diagnosis in. I think many of those are probably false negative tests. So I think with one of the lessons of our diagnostic survey really is that we need to use this technology and target the right patients. I think that massively parallel sequencing will be most valuable in those with genetically heterogeneous disorders, things like long QT. Those with enigmatic conditions and clues to suggest this is primarily genetic. It's not gonna be particularly useful in your 62-year-old smoker who had an MI, right? We kind of know why that person had an MI, but it will be useful in some subset of cases where we see a family history, where we see children with multiple malformations. I mean, the reason I'm intrigued by the fact that we had a 2/3 negative rate, which I think means we're missing something in children with birth defects, is that there are few things that are more genetically driven than our morphology, right? We look like humans because we have a human genome. If we had an earthworm genome, we'd look just like earthworms, right? So when you see something that disrupts morphology, there's a good chance that it's genetic. Now, I think recent events have shown us not to get too carried away with such reasoning. Look at Zika. When I first heard about Zika, I thought, wow, microcephaly, I'm used to thinking about that as a genetic disorder, but we have to remember that not everything is genetic. So I think that massively parallel sequencing of genomes or gene panels will benefit a subset of people, but it should be used like other medical tests. We don't just get MRIs on people willy-nilly just because it's a cool and powerful technology. We do it when there's a clinical indication, and moreover, we don't do a whole body MRI when somebody comes in where we're concerned about a brain tumor, right? We target that and we seek the information that is most likely to explain the patient's condition. So, this principle of thoughtful application is even more important, I would say, as we begin to envision the use of genomic technologies in ostensibly healthy people in the general population. So, I'm interested in how to use these tools to improve the health of the population. For some obvious reasons, right? The most obvious gains in medicine, in the entire history of medicine, have been in the context of public health. You have the chance to benefit millions. It's a lot better to prevent something than to cure it after it's occurred. And we have some wonderful historic examples. Vaccines are probably the single, maybe my candidate for the most important development in medicine ever. Fluoridated water is a pretty good one, right? You go to a developing country where they don't have fluoridated water and good dental hygiene. There's a lot of suffering from that. The problem is that public health involves a real different set of challenges and rewards than clinical medicine does. This is the world's happiest colonoscopy patient. This is Katie Couric, who to promote colorectal cancer screening got her colonoscopy on national TV. And the thing we have to keep in mind is that when we take a public health approach, we have to be very cautious about inflicting a population with our version of medical care. Partly because healthy people have less to gain than sick people. They're already starting out healthy, right? You can it seems like, only make things go downhill. There's a different relationship between the provider and the recipient. When I see a patient in clinic, they sought me out. That comes with a kind of implied contract that they kind of understand the medical system and that I'm going to do my best, but I might hurt them and I might not come up with anything. But when we go out to you and say, you need this, we better be very certain that what we're doing is a good thing. In part because the benefits are less obvious. All we can say is, great news, you didn't get sick. I mean, how many people have heard here the very reasonable objection that, well, I didn't get the flu last year and I didn't get a flu vaccine, right? It's a statistical argument that public health interventions work, and statistical arguments are a little opaque to a lot of people. And yet the downsides are easy to see in everything we do in medicine. Everything we do in public health has downsides, right? You don't get a free lunch. We're always going to harm people at some level. Moreover, we implement such applications en masse. It's not a one-by-one doctor-patient relationship, and that means everybody has a say, including idiots, okay? So who knows who this is? Jenny McCarthy, right, who has done a great deal of harm in arguing falsely, with no good data, that vaccines cause problems, undermining one of our, really best tools to keep people healthy. But that, people are going to have a say. That's, if you live in a free society, that's fine. People have a say. But what that does is it makes policy about such interventions much more difficult. And my conclusion from all of that is that the ratio of benefit to harm has to be very high before you implement something in the public health realm. Well, what about public health genomics? What are the right nails for that? So far, up to fairly recently, the field's focus has really been on common diseases with the hope that, if we can just nudge the needle with hypertension, diabetes, cardiovascular disease, we can do good things. And because even small progress with a common disease will have a good effect. But the hope that we can use genomics to assess risk and then predictably alter that risk has really not worked out very well. Applying genomic tools to common diseases has some real problems. One is the genetic influence in common diseases is just one of many things, right? If you look at heart disease, there are lots of reasons people get heart disease, and genetics is just one factor, and that puts a ceiling on the utility of genomic risk prediction. Moreover, the relative risk is quite small for the vast majority of genetic factors in common diseases, and I don't really know what to do clinically with a relative risk of 1.5 for most conditions. Our tools are blunt for intervening in medicine. And that means that a relative risk of 1.5, you've got to be very hesitant before you use that. Simply supplying people with their risk intervention or risk information does not seem to alter their behavior. And we've shown that over and over. And that isn't too surprising if you sat across from people and tried to get them to quit smoking, for example. My favorite, though, is confusing relative and absolute risk. I know what you're going to die of, right? And I would include myself in that too, unfortunately. We're going to die of cancer or heart disease. Now, some of us may get lucky and get shot or run over by a bus, but most of us are going to die of common diseases. That's why they call them common diseases. So for a common disease that you are already at a high absolute risk for, is it really very meaningful to tweak your relative risk? Lots of people die every day of heart disease who are at relatively reduced risk for heart disease. In addition, everybody's gonna benefit from population measures to reduce, say, cardiovascular risk and cancer. This brings us back to the same message, and that is that risk assessment will be most valuable when the identified risks are high. So that starts to make you think about what a productive approach might look like for genomic public health. Here are a list of a few diseases that are really readily amenable to prevention. Say, take something like Lynch syndrome, hereditary colorectal cancer. And let me ask you, if you carry a mutation that essentially guarantees you will develop a serious condition, say in the next 10 years, that can be readily prevented, would you want to know? Who would want to know? Okay, who would not want to know? Every once in a while, somebody raises their hand and says, I don't want to know. I always want to catch that person afterwards and ask why, but that's perfectly, reasonable. We should get to decide what information we want. But by that show of hands, you can see that there's a compelling argument to try to find things that are readily preventable. And I think that in there lies our most promising current application for genomics in the realm of public health. About 1% to 2% of the population carries a pathogenic variant that leads to a high risk of preventable disease. Lynch syndrome is a good example. About 1 in 400 individuals in the US population have a Lynch-associated mutation and are a high risk for colorectal cancer and some other cancers, many of which are preventable. But think about how we diagnose Lynch syndrome in 2017. We wait for the person and or a bunch of family members to get cancer and maybe die. That is a horrible way to make a diagnosis. We now have the technology to pick those people up. And I think that carefully applied massively parallel sequencing could allow us to sequence in a population selected genes that, when mutated, result in high risk of a preventable severe disorder. And many of those are cancer, but not all. There are cardiovascular conditions, etc. And that could potentially benefit 1% to 2% of the population, which is a pretty big number. And at first, the notion of public health and rare disease is a little bit, seems paradoxical, but think about probably the greatest triumph of genetics in the public health sphere, certainly, is newborn screening. it's predicated on targeting just the kinds of things we talked about, things that can be severe, that have a high probability of happening, and yet for which we have effective prevention. And you're all familiar with the Wilson and Younger criteria. So, I think about doing this, that's me as an infant with one of my DNA ties. I think about doing this in the adult population as kind of newborn screening for adults. And the beauty of the notion, I think, lies in part on the fact that in the US, it's about 0.12% of newborns who are found to have an actionable condition. And yet the prevalence in the adult population is an order of magnitude more than that. So I think that it's worth studying that. I think we have to select the right genes to target. Now, most of the 22,000 or so genes in the human genome are horrible candidates for screening. Most have no known disease associations. Many confer only modest or mild risk. Some show strong associations, but if you don't have prevention to go along with it, who wants to go screen for those? I think the idea would be to determine those few genes that, when mutated, really fit those essentially Wilson and Younger criteria, using evidence-based criteria, using existing guidelines from professional organizations. It would have to be iterative because the landscape changes. But we've done this before, right? We've done it in newborn screening, and I think we can do it in the adult landscape as well. And indeed, ACMG, ClinGen, and others have efforts underway to identify the most promising genes. We have a pilot study that's run by my friend Gail Henderson called GeneScreen, where we've looked at the feasibility of this on a very small scale. We have a very broad team from philosophers to medical geneticists to informaticians, where the intent is to recruit about 1,000 people over 4 years and screen them for a set of genes that we took a long time to kind of winnow down. And you can see that a lot of these are cancer genes. Some of these are, more generalized things like hemochromatosis. Some of them have to do with heart disease. And our preliminary results are interesting. 260 individuals have thus far been screened. And we have 3.8% positive, which is a little higher than you might think. And I think that says something important about what the penetrance of these conditions are when you ascertain people in an unbiased way. And you can see we've had a smattering of interesting results. This was a very recent one in which this woman had her thyroid out because she screened positive and had organ-confined medullary thyroid cancer, which was an anxious but ultimately exciting result for us right before we knew it was thyroid confined. That was anxious. As I mentioned, the increased prevalence over population predictions, I think, says something that we have to be careful of about our notions of penetrance. But I think we need to study this. I don't think we should just do it, because if we just do it, and my colleague Mary Claire King, I respect immensely, but I don't think we should just jump into screening every woman over 30 for BRCA1 and 2. I think we have to figure out how to do this best, how much it costs, etc. Otherwise, we're doing a large, uncontrolled experiment in public health, and the uptake will probably be by a very, very narrow, non-representative segment of the population. But if properly pursued, could really be a nice part of this notion, exactly whatever it is, of precision medicine. And I just like this picture because he's my favorite president, and because it's the first time that a State of the Union address was given with a DNA double helix beside it. So I want to just, in the last few minutes, talk about a few of the significant challenges to appropriately using genomics, both in the clinic and in the public health realm. And the biggest one is insisting on and generating evidence. This is just a long list and a very incomplete list of where we have gone wrong in medicine, but when we have prematurely implemented things that sure seemed like a good idea. From IV lidocaine in individuals who are post-MI to reflexive hormonal replacement therapy, we have continually, and it's kind of embarrassing as a field, that we continually be surprised that when we actually do the right studies, sometimes our ideas have actually hurt people instead of helping them. And I don't think we in genetics should be in a hurry to replicate this kind of activity. Yet we oftentimes, I think, are in too much of a hurry, and I think that can be laid at the feet to some extent of exaggerated expectations and claims. It just drives me nuts to hear people say, that soon everyone will have their whole genome sequenced. I don't think that's true. I think if that's the case, it's been a triumph of marketing over substance. That claim or recommendation is often predicated on the fact that it's cheap, okay? What that reminds me of is one of my favorite personal finance aphorisms, that an elephant for a nickel is only a bargain if you need an elephant and if you have a nickel, right? Just because something's cheap does not mean you should do it, right? I can get a Hello Kitty lunchbox on eBay for like $2, but I don't really want one. And moreover, even if it was free, low cost is an illusion in the medical system because misapplying a medical test is very expensive, both in terms of money as well as morbidity and mortality to individuals, even if that test is non-invasive, because downstream you ultimately get to invasive things in medicine. I just don't think healthcare systems are well served by routinely seeking vast amounts of information that we don't understand. Most of us have boring genomes, and that's a really good thing. And the parts that we do understand, well, we should think about maybe targeting those and investigating them. Not doing so begs for overinterpretation and harm, and we have to be guided by evidence in the clinical realm. I show an MRI again because I think we should treat genome sequencing and panels like we do radiology. We don't just, willy-nilly get whole body MRIs on people. I kind of have covered that, but I want to just stress the fact that we have to be cognizant of our ignorance. The associations even between phenotype and mutations in a given gene remain speculative to some extent. Our interpretive abilities remain poor, and here are two articles that really demonstrate that. Sometimes the press, especially in this last one, did not really communicate what the article clearly says. But what the article clearly says is, when we go do whole genome sequencing on apparently healthy people, we get lots of results that don't seem to mesh with what we know about pathology, and we have to be careful. Over-interpreting variants is one of the big hazards, I think, and I think it's the most serious analytic concern in clinical genetics and leads potentially to a crippling number of false positives. The point is that we make very serious, high-stakes decisions based on having called a variant pathogenic or not. We subject people to years of invasive screening or risk-reducing surgery, or we forgo such necessary interventions in their relatives if we misclassify them because we have misclassified a variant. We make important decisions about family planning, about abortion, and the challenge is that as humans we are evolutionarily predisposed to discern patterns even when they're not there. Right? We have all seen the man in the moon, but there isn't really a man in the moon up there, right? That's a trick of our brain. We evolved to see patterns in things, and it's been one of the reasons we're a successful species. But evolutionarily, seeing a false pattern wasn't a huge risk usually. You get a false positive and you swing around, and it turns out it's not a predator, right? Well, you can tolerate that level of false positives. But in the genome, it's so big it guarantees amazing coincidences. I don't want to end late, so I'm not going to tell you why I have a picture of Kevin Spacey there. So the bottom line is the bar for calling a mutation deleterious has to be very high. And that's critically amplified in the setting of applying this kind of thing in the population where the prior probability of a person having a disease associated with that mutation is low from the get-go. The role of the market is a vexing one in modern genetics because there's a lot of money involved. And I've thought a lot about this, and I absolutely feel that the market has a valuable place. And the place where the market is valuable is in the development of new technologies. One of the reasons we have massively parallel sequencing is because the market was a big factor in propelling that. But its role in proper application of such tests is really, really problematic. I don't think that we should really listen to those who argue that we should do a test because their stock prices will increase. We should be led by evidence of benefit, and that should be the single arbiter of whether we implement tests in medicine or not. I don't think aggressive marketing by the purveyor is the best way to drive deployment. And the point is, it's not a libertarian issue. We all have a stake in seeing that medical tests are properly applied, and that's because we all pay for each other's medical care. And therefore, if you have a test that is an inappropriate test and leads to downstream costs, I'm paying for that. So you know, it's not just a situation of it's my genome, I want the answer. We all have a stake in people getting the right test, and that's because everybody has medical insurance. Even these guys like Warren Buffett and Bill Gates, I bet you anything, have medical insurance. I would really recommend this commentary by Kim Rockwell, that makes this point much more eloquently than I do. One last audience participation slide. Here are some Mendelian diseases like early onset Alzheimer's disease, fatal familial insomnia. If you harbor a mutation that essentially guarantees you will develop a severe, untreatable neurological disease, say in the next 10 years, would you want to know? So who here would want to know? Who here wouldn't want to know? Who here isn't sure? So it's like in my scientific survey here, about 1/3, 1/3, 1/3, right? The point being that nobody's right or wrong about that, right? You know, you can't gainsay somebody else's choice. We have to learn to navigate that value heterogeneity, and we need to understand how to communicate this to people so they can make the decision that's best for them. Politics is a challenge to precision medicine. Prevention is worthless if the population doesn't have access to that prevention. So I don't care which side of the aisle you're on. If you believe that prevention is important, then you got to figure out how people can have access to that prevention. And that's a major challenge, especially right now. I'll end with just a few more random challenges. Genetic discrimination, you know, we have protection for now against healthcare and employment discrimination, but it sure doesn't include life insurance, disability, long-term care insurance. I used to think this was a joke, right? I mean, I put it in because I thought, maybe allelism, right? But the CEO of 23andMe said in the Washington Post, we envision a new type of community where people will come together around specific genotypes, which I found kind of chilling because I think we've been down that road before. Gene patenting was largely put to rest by the Supreme Court in June of 2013. But, you know, we still face problems with proprietary databases built from people who, you know, they didn't want to have genetic testing. They got sick. They got cancer, for example. And now somebody's, you know, keeping their data secret for gain. I think that's a problem. Privacy issues are a problem. I like Scott McNeely's comment that privacy is dead, deal with it. I think it is kind of dead, but we have to deal with it. And finally, the last thing I'll mention is, you know, I don't want to use too much hyperbole, but I think that with a lot of technological advances like CRISPR-Cas9, we really are on the threshold of being able to manipulate the genome of an embryo. And when we do that, we have to remember that we are manipulating the genomes of countless generations in the future. And we're not, you know, we are going to have mistakes. We're going to have problems because we never do anything without screwing up sometimes. And we have to think seriously about that. Genetics has brought us to the cusp of power that is kind of unimaginable, and we need to take that seriously. So I'll end by thanking the patients and the subjects who have, you know, been so instrumental in studies, and all of my colleagues and you guys. Thanks. So I don't know if there's time for a question, but
Thank you, John.
you bet.
A very sobering and enlightening talk. We do have time for questions, John.
So, I really enjoyed your talk, and variants of unknown significance are being discovered at an alarming rate. And there are a number of instances where people have assigned pathogenicity to variants where that's not true. So, one, do you have any idea how prevalent that is? And two, do you have any ideas for suggestions of how to clean up the literature when that happens?
Yeah. I wish I could fix it. As far as prevalence goes, I mean, just the frequency of VUSs, as you know, is going to be in the double-digit percentages, you know, anytime you do a panel, right? I think, I mean, I hate to use platitudes. This sounds like a platitude, but I think the only solution is education, all right? We need to educate people that VUSs should be innocent until proven guilty, that they should not, that there should not be changes in medical care based upon them. And that's a hard sell because we want to find answers, we want to find patterns, we want to ascribe causality to things. And, you know, my own rule of thumb is when we're in sign-out conference and we've been discussing a variant for more than like a couple of minutes, it's a VUS, right? If we've had to discuss it that long. There is one possible solution, and I hesitate to say this because it sounds like I want to feather our nest as medical geneticists, which, believe me, is not the case. You could, I was just talking with Jeff Ennis earlier, when I was an intern, the only way you could order tobramycin for a patient was if you were on the infectious disease service. I did not think it would be unreasonable to say, look, you want to get a big panel, you want a whole exome, involve a genetic counselor and or a medical geneticist. I think that, you know, nobody's going to let me, for very good reason, do, you know, a knee arthroplasty, right? Why should we, this notion that everybody should be ordering whole genomes, to me, is silly. It's, if there is a role for the medical geneticist in modern medicine, this is it, right? Navigating panels and whole exomes. So I think those are the education and maybe limiting who can order it. So, yeah, sure.
I think your point about the issue of prior probability is really important and brings to mind a paper that Les Biesecker published a year ago or so where he actually went back through the West data and then called back patients for their NIH study and found out that in deep phenotyping they could find that the prior probability may be as high as 1 in 30 individuals rather than the abysmally low number we tend to think of when we're talking about this kind of a subject. What do you think?
So the trouble I have with that notion, if I'm remembering that paper, is this notion of kind of aggregate prior probabilities. And the reason I have a problem with that is it's kind of like, to me, saying, I'm going to do 2 tests in you. One is looking for fatal familial insomnia, which is about 1 in a million, and the other is going to look for factor V Leiden. Not that you should look for factor V Leiden, right? Which is about, what, 7%. And then aggregating those and saying, oh, the chance of a positive is 7% for a positive test on this panel. And there, so I actually think there's a problem with the calculation there. I think you need to use strict and the best population frequency for each entity that you are screening for. I didn't understand that paper, and maybe it's my own understanding, but yeah.
Thanks a lot, Jim. Very exciting. It just occurs to me that one solution to the VUS problem is really more sequencing and getting more data on who has the variant. So that goes counter to.
But you know, I didn't make myself clear. All right, and where you bring up is what I need to make more clear when I say that. I think we should sequence the hell out of a zillion people, but in the research arena, right? You don't dump that just because we need we need more people imaged too to figure out. But you don't go out and just, you turn that over to the clinical enterprise and just order needless tests on people, right? What you do is you set up a big research cohort, like maybe the All of Us cohort, and I want to see 1 million people research because you're right. Now, even sequencing everybody in this country will not solve VUSs, right? How many years now are we post BRCA1 and 2, and yet, the incidence of VUSs has only gradually declined. So the genome's big enough and the combinations of problems are high enough that it won't solve our problem, but you're right. And I'm glad you mentioned that, made that comment, because I don't want anybody to think that I'm not advocating for lots of sequencing. I am, but not under the pretense that it's good clinical care or should be paid for by third-party payers.
So not to belabor the VUS point, but at what point can we say that we've achieved the burden of proof, right? Because we can't do functional studies on every mutation or variant that we find every VUS. So for example, if we find a VUS in a child with intellectual disability, it comes back to the clinician to decide if the clinical information actually fits that particular gene and the phenotype that we would expect, and then also doing additional family studies. So for example, what I worry about is people saying, well, it's just a VUS, it's not important, and why are you studying this? When the clinician puts together the phenotype data and the family studies, actually at some point you're going to reach a level of significance that convinces you. And I know that there's the ACMG guidelines and everything, but I guess I'm just saying, at some point we have to cross that threshold.
I couldn't agree more. And what I would say is there are some very straightforward tools that help, right? For example, population frequency, right? Whether it's de novo or not. Maybe the most powerful of all, but you just don't have access to it oftentimes, is segregation data, or you don't have access to it in a statistically significant way, right? We have to combine all those things. I'm intrigued. I don't think it's going to solve all our problems, but I'm intrigued by the notion of massively parallel functional analyses. The problem with that is you need some insight into the biochemistry, and beyond that, even if we think we know what a protein does, it may have other functions that our functional testing misses. So it's not going to be the whole answer. So I agree. And what we have to do is we have to use kind of a point system and add them up. And I guess my major message is we need to have a certain robust skepticism about being convinced, because I would argue we're too easily convinced of causation, and we need to guard against that.
So sorry, you hit a trigger with me on the million-person project. So given benefit versus outlay of costs, and that money's not endless, do you really think sequencing 1 million people is going to give us more information than funding individual R01s or continuing on mechanistic research? Because I have some issues with that.
I don't. Okay, and I'm trying to make lemon out of lemonade. All right, I think that there has been money put into that effort that's going to be spent on it. I think that we can get something good out of it. But if I were heading up the NIH, God forbid, right, or the HHS, no, I wouldn't put those resources into that. I actually have profound questions about whether it's actually going to answer much at all, because I think it's going to be very, very difficult to do, given the kind of constraints we have, given the society we have that is fixated to some extent on privacy, where medical records are not very easily obtained. I'm a skeptic about the All of Us cohort. My advice, which I don't think anyone will heed, in a couple of things I've written in the New England Journal and in the journal I edit, has been, well, at least if you're going to do this, pick some low-hanging fruit, for God's sake, right? Identify people with familial hypercholesterolemia and Lynch syndrome and see if you can, right, benefit them. But I'm a skeptic. I'm in your camp on that. Yeah.
Jim, this is very nice and actually really thoughtful discussion. Your study where you're looking at individuals for variants, perhaps in the 56 genes that, are secondary
much smaller,
findings. So even smaller.
Much smaller than 56. It's 17 genes for 11 conditions, and I thought it should be like 15 for 10 conditions.
Well, the study itself prompts the question of whether one could envision a clinical test which could be used as an adult screening. Yeah. And what, in your opinion, would it take for you to support such a clinical test? What level of evidence would you want to see?
So I just wrote the best grant I ever wrote that didn't get funded that answered that question, okay? I mean, it's the best grant I ever wrote, and it didn't get funded, not the best grant I ever wrote that didn't get funded. And here's what I think we need to do. We need to do it on a large scale because the number of positives, even at 1% to 2%, which is a great figure for a public health intervention, but even at that, you need a lot of people to get enough positives to study. So you need to study 20 to preferably 50,000 people, number one. You need to do it in a healthcare system where you stand a good chance of getting follow-up. And your immediate or your near-term follow-up have to be proxies for clinical utility, like did people adhere to the recommended actions, right? Like did they get colonoscopies? Did they get MRIs, right? If it's BRCA1 or 2. And what the findings were on those things. And then ultimately, if we did a lot of power calculations for whatever that's worth. And I think that in a 4 to 5 year span, you could derive good evidence of the costs and the utility with intermediate measures. And it would probably take you 10 years, but you'd have robust power with these because the outcomes are fairly common, right? The bad outcomes. Within 10 years, you'd have good power to be able to detect really hard outcomes like death and overt appearance of cancer, cardiovascular events, etc. So I would, it's tricky in medicine. If you delay too long the implementation of a good idea, you penalize people, right? And if you do it prematurely, you penalize people. So I'm not saying we need a 10-year study that ironclad shows clinical utility for something that's a good idea. What I'm saying is let's cautiously implement while really setting up the infrastructure to study it, and then study it. The problem is we're not studying it. And if you look at like Color Genomics, which I think is a good lab, right? They have basically a marketing campaign to do their cancer panel as a corporate perk, right, for executives, an extension of the executive physical. And, do I think there's promise in selected screening? You bet, right? Hopefully you could see that by my talk. But do I think we should just do it? No, I think we should perhaps study it, right, and cautiously do it while we're studying it, right? I don't want to be in the camp that, oh no, we need 10 years of clinical utility data before we do anything. That's not sensible, right? But on the same token, you don't just do stuff because it seems like a good idea. Right? The stakes are too high. So. All right.
Jim.
Yeah.
The current theme is about patient-centered care, respect for the patient and the family in a market economy. So the topic that triggers my question is the publication of the book and now the movie called The Immortal Cells of Henrietta Lacks. Yeah. In fact, it was shown here to our graduate students last month, and it's the subject of a showing from the Public Health Genetics Genomics Institute of the University of Washington next Thursday. They put it online in one of the biggest halls at the University of Washington, and the place was filled online with everybody reserving seats. The question of that movie, I think, although it's a little complicated, and it's from 1950s for God's sakes, is who owns tissues and who owns
Yep.
the rights to benefits from the tissues, including the economic benefits. You thought about that at all?
Yeah, and it's not that I claim any special expertise on it, but sure, I've thought about that. I think that, to me, there's two really important issues that we should separate. One is egregious harm and disregard for harm to and disregard for individual research participants, which is inexcusable. All right? And I think, for example, about the Moore case that went to the, I think the Moore case is the hairy cell leukemia case, where the person was brought in over and over to get biopsies and all because they took out his spleen basically for research purposes, never really told him what was going on. And it's inexcusable to keep people in the dark, to pursue research, regardless of whether there's monetary incentive there or not, without participants being true partners in what you do. So that's one thing. The second question, I think, is much more subtle about who owns the material. I would tell you that I suspect that the value of any of our participation in a research study is actually very small and very incremental. And I don't think it is either worthwhile or particularly useful to turn research into the notion that somebody is going to make a lot of money, because I think the vast, vast majority of cases, the individual participant is not supplying uniquely valuable information, right? It's an aggregate kind of approach. So I'm not really, and moreover, I think that if we start turning research into a monetized transaction, we undermine the utility of research. I'm so impressed by the people who have taken part in our studies who over and over and over say to me, I'm in this because, maybe I can help somebody. There's a real altruistic bent, and I don't want to squelch that by monetizing it. So I guess my feeling is, I don't like, as you can probably tell, I don't like the influence of the market in the implementation of scientific discoveries, et cetera. I recognize that these things end up being valuable, but I would actually tell you that the institutions that support research, that support researchers, they are the ones who are, in general, the ones who kind of own the rights to that. And I don't think it's very workable to really hold out what I think is a false hope to participants that they're going to make money. The case of Henrietta Lacks is a very unfortunate one. But I think that the solution to that is really transparency, making an effort, a real effort, to make participants partners in research. And again, not holding out the false hope that everybody's sitting on a gold mine because they have some biological disease or a trait. That'd be my thoughts. Yeah. All right.
Jim, let me just thank you again for a very stimulating and provocative talk. It reminds us that being a physician means making decisions based on inadequate data when it isn't there. And I guess the other is it reminds us of our huge amount of ignorance about the workings of human biology.
Yep.
And that as much as we keep learning how much we don't know and how difficult it is to make these decisions. But I think this is a very provocative.
Well, humility in the face of our own ignorances, we should always retain.
We should try.
I tell the students, if you aren't comfortable being humiliated by your ignorance every day, pick another field, because medicine is not for you.
Right. So thank you very much. Thank you all for coming. There is a reception with refreshments across the hall. Invited to join us.
Yeah.